Hi, we're gonna be doing chapter 5 and chapter 8 study review. So we're just gonna be having a conversation. Here we go.
Yeah, the chapter is the structure and function of large biological molecules.
Yeah.
Alright, so the basics. Adam, take it away.
So a monomer is a set of building blocks that make polymers, and all living things are made up of macromolecules, which can be divided into carbohydrates, lipids, proteins, and nucleic acids.
Mm-hmm.
So all of these can be polymers except for lipids because they are
Hydrophobic.
hydrophobic.
Which means that they can't mix well with water.
The opposite of hydrophilic.
Philic.
Yeah, so examples of hydrophobic substances would be like oil. I'm sure everyone's seen like when you pour oil over water, something they don't mix, it just floats on top of it.
Wax.
Yep.
Ooh, wax. Yes.
And
Very
then,
true.
what would be an example of hydrophilic?
Mixes well. Wouldn't it be like sugar?
Solvents.
Salt.
Things
Solvents.
that, solvents. Anything that can dissolve into water. It doesn't mix well.
Oil.
It goes above it. I showed
What's on the
oil,
top?
but that would be. Oh yeah, it sits on the top.
Yeah.
It's hydrophobic. Y'all
We
get
know.
it.
We know.
We know. Another thing, like anything that you use in like mixing, like for like if you do those little like energy drinks things, if you pour those in, those are like really good.
Example. Yeah.
Pre-workout.
Oh yeah.
Yeah.
Pre-workout.
Pre-workout.
Adam has some experience. Some experience in that realm. What else? Oh, so since Adam did mention polymers, the smaller version of that would be a monomer. It's the building blocks that make up
The
polymers.
polymers. Yeah.
The monomers have like little specialized units. But they like repeat throughout the thing.
Specialized units, yeah, they're building blocks.
And then the reaction that actually creates the polymers out of the monomers is called dehydration reaction.
Which does Adam.
They're connected by covalent bonds, and any polymer is 2 or more monomers.
Yes.
Yeah. Adam also, he has the dehydration. Could you read out?
Yes.
Connected by reaction in which 2 molecules are covalently bonded to each other with the loss of a water molecule. This is known as dehydration reaction, which is basically what they're talking about.
Yeah, it's where to get 2 monomers together, or 2 or more monomers together, you have to take out water.
Yes.
Which it's like, I'm trying to think of an example.
I don't know. That's basically it, like H2O.
Yeah.
But the adverse, the coinciding part of that would probably be the hydrolysis. That's how it breaks it down. So you're actually, which it seems backwards, but you're actually adding a water molecule
The reverse.
in order to break it down. Which is what happens when you like work out, basically. Adam would know more about that. You know, when you work out, you lose those calories. You have to
Here's a
raise the Celsius.
drawing of the
Closer.
dehydration reaction. And as you can see within it, it's a little bit reversed as of now, but so the H2O molecule is actually leaving that bond. It removes the water molecule, obviously, and it's forming a new bond of its own. Whereas with the hydrolysis, it'd be adding that H2O molecule within that, and then it would be breaking the bond of the H and the HO by itself. So that's what that would look like.
So, sugar. Carbohydrates, which is basically just sugar.
It's the
Includes
polymers of sugar.
Polymers of sugars.
Yeah.
And one other word for sugar is like monosaccharides, which
That's
is
the
the
simplest.
simplest, like one molecule of sugar, if you will, is a monosaccharide.
Whereas a polysaccharide is like more.
Complex.
It's obviously it's more
Yeah, or
complex.
the disaccharides. Those are the ones using 2.
Yeah.
Yeah.
Good way to know if it's a sugar is if it ends with ose, right?
Yes.
So glucose, fructose, ribose. Right?
Ribose.
All, yeah, they're all.
Yeah, they're all sugars.
Sugar.
Sugar if you want.
And then like whenever you break down a polymer into like the monomers, like where does it get released to?
The monomers get released into the bloodstream.
Nice.
Hey,
Hey, he really out there.
Everybody with diabetes, pay attention.
At me.
Listeners out
That's how
there,
it's
like,
supposed to work.
yes.
Unfortunately, it doesn't work. Sorry. Sucks.
Relating it to real life.
Yeah.
Polysaccharides are a form of macromolecules that have a few thousand, actually, monosaccharides. They're relatively large, and they're also called
Glycosidic
Glycosidic
linkages.
linkages.
Yes.
Wouldn't that be the same thing as starch?
Well, starch is a storage form of it. Yeah.
Yeah.
So in like plants, that's what they used to.
Oh, plants store starch.
No, plants use starch
Starch
to
to
store
store.
things.
To store
Oh.
glucose.
Yeah, like
Yes.
and it's in their chloroplasts.
Okay.
A little.
And then,
A little.
well, that's not the only thing polysaccharides do.
What else do they do, Meg?
They have they have hydrolysis in them, and then they also are there for protection of like the plant if it were to like go through a drought. It's like your protection.
So like during the winter, basically. Right?
Yeah.
Like the hibernation season.
Yeah.
So what happens if the winter is longer than expected?
Well, what does happen?
The starch.
Yes.
They're not able to
Yeah, what if they don't
store enough, and then
Die.
they die.
Tragic.
That's like when the winter's really long, the sun hasn't been out in a while.
Like the trees.
Yeah, the trees
The trees
Oh, yeah.
store.
don't come back to life.
The
Yeah,
roots just kind of like give everything that they have, and then they just fall.
they have nothing left to keep going.
Right. So we know that plants hold starch, or at least that they store starch, in order to hold glucose and everything. But we also know that animals store glycogen. So what do animals use this glycogen for is my question.
So to store polysaccharides, like the same thing for like.
I know, but like,
wouldn't it be like the same use as like plants use them, but it's like for animals, I guess?
okay, but how so? Like, what are they?
Yeah.
What exactly are they using the glycogen for? Is my question.
It's like
Consumption,
consumption, yeah.
food,
Like
breaking down
Releasing the
food.
sugars
The sugars for food.
at certain times.
Yes,
What?
as energy.
wouldn't it help like whenever you're in like a cold situation and like all the storage of it can like be released for like heat
Mm-hmm.
Yeah.
purposes.
replenished by consumption of
And-
So glycogen stores are depleted in about a day unless they're replenished by consumption of food. Oh, so is it kind of like a last resort type of thing?
I think
Like
so.
it
Like
keeps them.
I don't
It's
think
like
it's
a
not-
backup?
It's like a constant thing.
Oh.
Yeah, that's a
But
constant
it's like-
thing.
It's a constant. It's always working.
A
That's
backup
why
thing-
we
a
always-
backup thing would be
that's
like
why
fat,
we always
right?
eat, and then
Okay.
fat would be
Fat
the
would be the
burning of
backup.
it as the last resort.
Okay.
That's why when you work out.
So, other than the storage polysaccharides, there's also structural polysaccharides. Saccharides. I don't know if I'm saying that right.
Saccharides.
Saccharides. So they're kind of the building blocks in this situation under this, I guess, subsection. And one of these is cellulose, which is, well, obviously everyone knows that plants are the only ones who have cell walls.
Mm.
And
And
that
cellulose
is,
is the components of the walls.
yes, so it does make up a large portion of that.
is there something that helps in the structure of animal cells, or is it just the plant cells with their walls and like animal cells are just like have fun?
No, that'd be inside.
I have no idea. Well, because
Nucleus is the
nucleus is the
inside.
inside. It's
Yeah.
a cell membrane. And then
Yeah.
Cell membrane, that's
I
what
don't
it
know.
Yeah.
is.
I don't know what makes up the cell membrane.
Here.
Probably
So-
proteins,
Yeah,
because that's the
probably.
answer to everything.
We got you, Miss Gaston.
we listen. Proteins.
We listen a little.
So,
Speaking of proteins, like enzymes, bruh.
I
Like-
guess. Oh, an enzyme. Well, we'll let you know what an enzyme is.
oh,
Don't
an
you-
enzyme. I'm glad
don't
you
you
asked,
worry.
actually. Enzymes,
They're specialized macromolecules that speed up chemical reactions. Specialized proteins, to be exact.
Protein's
Oh,
the answer.
Yeah.
Yeah,
Yeah.
similar to
Proteins
that.
are the answer.
Similar
So-
to that as a catalyst, which
Yeah, it's
speeds
a
up-
catalyst.
it speeds up chemical reactions. It's a very common.
Although enzyme, what's the difference between enzyme and catalyst?
A catalyst, like
Is a catalyst a protein?
it's any substance.
I don't know. I
that
thought
speeds
Yeah,
catalyst
it's any
was an
substance
enzyme.
that sticks
Yeah.
onto it makes it faster.
But-
Yeah.
It doesn't have to be
yeah.
necessarily a
up.
protein.
Other one's a protein.
But are catalysts enzymes?
So catalyst.
I think.
It can be. I
Or
think
enzymes
it
are-
I
can
think
be,
it
but
can
it isn't
be.
specifically.
Yeah.
So I thought
Okay.
they were like the exact same.
No, they're not the
They're
exact same.
they're very similar, which
Okay.
is why I got confused on the last quiz.
Yeah, me too.
I feel like
Get that
everyone
question
missed
right.
that. For sure, for sure. Hi, guys.
So next topic, fats, if you will.
Lipids.
Lipids. Well,
Earlier we did mention that they're
Lipids.
hydrophobic. So.
yeah, we did mention they're hydrophobic. Mixed with water. Okay. The reason why they are hydrophobic is because the molecular structure, although they may have some polar bonds associated with oxygen, lipids consist most of hydrocarbon regions, which means they just don't mix. Don't know how to explain that.
There are two.
If you know, you know.
There are two forms of lipids. There's saturated fats and unsaturated
Unsaturated,
fats.
yes.
Yes.
This mainly has to do with the hydrogen that is in it.
Mhm.
If it's saturated, it means it's saturated with hydrogen, so it has more hydrogen molecules. Unsaturated has more than one double
or
bond.
more double bonds
Yeah.
with one fewer
But-
hydrogen atom.
Yeah, so it
Would-
doesn't have as much.
Would that mean that saturated fats are less likely to are more resistant to water, and the unsaturated are still resistant to water, but not as much as the saturated?
I guess you could say that. That is
Yeah.
more. I never considered it, but that makes sense.
And is it because of the double bond exactly, or is it just the fact that it's filled with the hydrogen?
I do not know.
Yeah, I don't know either. It's like-
Let's see.
We'll Google
Saturated
that.
fat
We'll
would-
Google
a
that.
saturated fat would be like butter, I guess.
Yeah, it says that it'd be butter, hydrogen, solids.
Yeah, it's a solid. Your saturated fats are gonna be solid.
In a more solid form.
Yeah.
And your
unsaturated
unsaturated,
would be like oils,
Yeah.
liquids
Yeah.
and stuff. Liquid substances.
yeah.
Soluble bonds. Yes.
Hey.
Double bonds. Yes.
What's an example of an unsaturated one, Adam?
An unsaturated fat?
Yeah, like it's liquid form. What's a liquid one?
Like butter?
No, that's a solid.
Oh, that's a solid.
Mhm.
A liquid fat, wouldn't that just be
Like
Oil.
oils?
Like a solid fat would be like fat, like animal
like
fat.
yeah, like body
Yeah.
fat.
Yeah, so that's your saturated.
Or
So an unsaturated could be like vegetable oil or
Yeah,
baking,
vegetable
like
oil.
literally any type of oil.
Yeah.
Okay. That makes sense.
So. Yeah, phospholipids. Take it away.
Phospholipids is a similar to a fat molecule, but has only two fatty acids attached to the glycerol rather than the three.
It's a huge part in cell membrane, so I guess we could probably thank them for us living. So
Yeah.
So
Shout
it's
out
a cell
to
membrane.
phospholipids.
Shout out.
Shut up,
Thank you
phospholipids.
guys. I really appreciate the work that you
Thank
do.
God.
Yeah. So
And-
so that's what makes up the animal cell, whereas the
The
plant cell is
it's made up of glucose.
of glucose.
But not glucose.
Speaking
Cellulose.
of
Cell.
protein.
The
Cellulose,
cellulose.
which is a structural polysaccharide.
Yes.
Yes, yes, yes.
Oh, we
We love-
data. Making connections.
So steroids. Steroids are lipid characterized by carbon skeleton consisting of 4 fused rings. So, isn't steroid a protein?
No. Steroids are lipids.
Lipids, right?
Lipids.
Yeah.
Which is also a type of, isn't cholesterol turned into
Yeah.
steroids as well?
Yeah,
Yeah.
which is probably like one of the more important or more well known
Yeah.
forms, especially since like there are so many people in this day and age who have problems with their cholesterol. That's because they are eating too many fats, like
Mhm.
greasy, unhealthy foods that contain a lot of bread, a lot of like breading and stuff.
Chicken Express.
Yeah, yeah. I mean, don't hate, but
So loud.
yeah, that's a lot, bro. Don't call me out like that, but Yeah, that is why a lot of people do have problems with their cholesterol. That later usually leads down the line to like health issues, heart attacks, strokes, and such because of clogging the arteries. A little extra information.
A little extra landing.
Yeah.
Eat your Cheerios. They're high in cholesterol.
Good cholesterol. There's good
Yes.
cholesterol and bad cholesterol. I don't.
What's the
Some people
difference?
are blown away by that, but
So like that's why they put the good cholesterol like in your butt, right? Like when they do
The shots?
the shot things.
Yeah.
Oh.
Yeah.
there is a difference. A lot of people think it's just like bad.
Really?
Yeah.
That's what I thought.
Why
too,
do you
'cause
why do you think cholesterol? You need it to live.
Yeah. But like there are
What's the
two.
difference between like the Cheerios cholesterol and like?
Like the Cheerios one is like
I don't really know what the difference. Is.
I think it's like it depends on like the amount.
We can look it up.
I think it's
So-
more saturated.
Ooh, it might be.
What? Whoa! Wait, let's look it
Whoa.
up.
Let's see.
Difference.
Don't you think? Because like, I don't know.
And good and bad cholesterol.
Yeah, we're gonna need to know this really.
Anyways, while she looks that up, let's move on to amino acids, which is an organic
Okay, I
molecule.
found it. Shut up.
She got it.
She got it.
Bad cholesterol involves LDL levels that are shown in a link to cardiovascular risk, which I informed you on previously because good cholesterol is known as high density lipoprotein. So I guess it's a protein in that
Yeah,
form.
that's what I thought.
It brings cholesterol from blood and blood vessels back to the liver. So it's the circulation part of it. It takes it away from your arteries, which is what we typically associate with cholesterol,
Yeah.
and it takes it to your liver. So, bad cholesterol, known as low density lipoprotein, circulates cholesterol into the body. That's why it's bad, because it puts it through your bloodstream.
Ooh.
So we learned something new.
Yeah. Okay.
For sure.
That makes things like a lot more like clear though.
Mhm.
Like I don't know, just knowing like more so on that.
Yeah.
Helps.
Definitely.
So amino acids, that's what I was saying.
Whatever.
Is an organic molecule with both an amino group and
Carboxyl group.
carboxyl group.
Yes.
What's the difference though?
Between what?
The amino group and the carboxyl group.
It's the difference of like what it's made of because each of them have like their own.
What structure?
Amino
Yeah, because
Yeah.
the amino
Chemical
groups
structure.
have hydrogen and oxygen, whereas the carboxyl groups obviously have
Well,
carbon, oxygen, and hydrogen
Hydrogen.
hydroxide, if you will. And here, I'm gonna actually make a little
a little
illustration so you guys can see.
so like
And
we
the
have to be
section-
careful when we say structure because something can have the same structure but have like different molecules and can be something
Sure.
totally
Oh, yeah.
different. So we have to.
We're
Like,
using the term very loosely.
yeah, we're using it a little too
Oh,
loosely.
God.
The AP examiner graders are not going to be okay with it.
Yeah.
Show
We're
my
sorry, ladies.
illustration of the
Show
amino
illustration.
acid, if you will, Meg.
Okay, okay,
And point out which side's which.
okay. This one's the amino side, and this one's the carboxide side, and like this bonds it together, so like that makes it an amino acid.
Yes,
Awesome.
Yeah.
for sure, for sure.
Okay.
Oh,
Emmy, we gotta do some work.
Emmy, Emmy, come
sorry.
here, come here, Emmy. Emmy, come here.
Okay, moving on.
moving on, ladies.
Hmm.
Now we're gonna jump into our polypeptides.
And those are amino acid polymers.
Yes, that is the other name for them.
Yes.
Yes. Okay, so where two amino acids are positioned so they are carboxyl group, one is adjacent with the amino acid to the other, they can become joined by dehydration reaction. With the removal of water molecule, the resulting covalent bond is a peptide bond. So what that means is
Wait, is that the
It bonds.
removal of just one of the monomers to the polymer?
yes.
Okay,
the
so-
H will.
So it leaves it, making it a covalent bond.
Yes.
So it makes-
Connecting them basically in the same process we talked about the dehydration.
Yeah.
And when
Okay.
done repeatedly,
So it's
it eventually makes up what we call a polypeptide.
A.
Showing us
Whoa,
minute.
so
A little
numerous.
smart.
So numerous
What can I say?
peptide bonds make a polypeptide.
Polypeptide.
Interesting.
For sure, for sure. Just keeps going.
So Ronnie, can you inform us a little bit about the different
Models.
models that are available to show this?
Oh, but of course, okay. So first you're gonna have your ribbon molecule.
Can you illustrate as well?
Oh yes, I may. Okay, so as I'm talking, I'm kind of copying the little diagrams on the book.
As we speak.
I won't be as good
As
as
we
the artist on here, but within the ribbon model, the single polypeptide chain sort of folds and coils within one another. So
It literally
It-
looks like a shoestring.
it looks,
like
It's
a shoestring.
kind of self-explanatory. I like
it
ribbon.
really does. It looks like,
If you've seen
ribbon-
a ribbon, you've seen a ribbon
and
model.
its
I thought-
delicacy, like how it folds over, it looks similar to,
Like if you scrunched up a shoelace or like a string of
yes, for sure.
Like a
spaghetti.
ribbon.
Yeah, like, wait, I got y'all.
Oh, thank
If
you.
you have a
If,
ribbon,
like,
then
you
have
take
it.
this boy and you,
Yes,
like, undo
for
it
sure.
and, like, just...
Put it in a
Scrunch
ball.
it up.
I'm
Alright.
not going to do
The
that.
second time is space
This
filling
is
model.
my knitting yard. I'm not going to do that.
Space filling model is kind of what you'd think of when you think of molecules, which is those big O's, like
Big, like spheres, little
Spheres, like little
Yes, it has like a
spheres.
globular shape.
yeah, like if you would write out a an element and like combine it like H2O, you would draw an H and two O's on the side. That's basically what a space filling model would look like, just a bunch of them clumped together.
It's thick and it has a little groove.
Every
We're
time
gonna-
I look at this, I always think of the Big Bang Theory.
Yes! Here, show them my illustration.
It's kind of the stereotypical idea
Yeah,
of
like-
a model.
Like
Yes, for sure.
Isaac Newton.
I think
What
by
Isaac
far,
Isaac
Isaac
Newton.
Newton.
by far the wireframe model is definitely the most unique. I've never seen
Oh yeah, it's like
anything like it. It kind of looks like, it reminds me of like barbed wire.
It's
And
kind
It
of
has so many.
Yes.
It reminds me of barbed wire because it has so many like pieces sticking
it
off.
still follows the structure like the ribbon model,
Yeah, it
but
does.
instead it like
It has little grooves coming out of it.
yeah.
And I'm gonna try and illustrate that within.
Well, like
I'm not no Picasso.
There's also
She
a target
tried.
molecule on there.
Yes. There is. There
This is
is.
target molecule.
The yellow or the orange structure that I'm gonna put in here is gonna be the target molecule, but you'll be able to see how there's little spindles on it.
What is the
Yeah.
target molecule?
Like if we're looking here, it's right here. That's your target molecule.
Yes, we got it. We got it.
moving.
All right, so we are actually gonna move on to certain diseases
that are associated
with
stuff.
structures and
Yeah.
such.
So one of them is sickle cell disease, which you've probably heard of, which is an inherited blood disorder that is caused by the substitution of one amino acid for the normal one at a particular position in a primary structure of hemoglobin, the protein that carries oxygen to the red blood cells. Basically, it means that like your red blood cells don't form correctly, and instead
They're a
are
strange shape. Yeah, they're
Strange
different.
shape.
They're like they've got like a divot in them, right?
Yeah.
a sickle,
then whereas
in-
As
a
opposed to a fully rounded.
round.
yeah, because they aren't.
This is like I'm pretty sure it's, correct me if I'm wrong. It's found most recurringly in like African countries where there's no malnutrition. and
Yeah.
stuff.
Yes.
I too.
Yeah. It's like it has a little divot or divot.
It'd be like a lack
Right
of-
here.
Yeah.
Like it doesn't have to be like right there, but like it.
Yes, of course, but of course.
Divot.
A divot.
So
Um.
denaturation.
Oh really? Why? Okay.
Sorry, my voice has gone a little faked.
So like if change to the protein and like unravel and lose its shape, it would be called denaturization, which basically means they loses from like its original shape, like where it's supposed to
That's
be.
kind of what's happening in sickle cell.
Yeah, because it
Wow.
goes from its original shape, which is
And then
supposed to
it
be a
goes
circle,
with the
and it
little divot.
deforms it, so it makes it divot. If you will.
Denaturation. Kind of crazy.
These. Oh, well, Oralia can tell us about this. This is how we are able to fully understand these
The
processes.
protein structures and such.
Yes.
Okay, so we're actually able to look into these certain diseases as well as like hemoglobin cells and different proteins with our new X-ray crystallography. This has been used to determine the 3D structures of many proteins, such as the ones that we've gone over. You can see how the shapes change, the denaturation with these X-rays, and that's scientists really utilize them.
Yeah, it's important. It's an important process in the scientific realm.
For sure.
Moving on. Okay, so now we're going to move on into what people most commonly associate with this chapter, which is nucleic acids.
Yeah.
Yeah. So there are two types of nucleic acids: deoxyribonucleic acid, which is it's more commonly known as DNA, and ribonucleic acid, which is RNA.
RNA. Our messenger RNA.
Yes.
Our messenger RNA that went to make the copy.
Yes. So they enable living organisms to reproduce their conflict component from one generation to the next. It's a unique string that makes up DNA. So everyone's pretty different. That's why everyone is so unique.
And then isn't DNA and RNA both have different letters?
That connect to them. That's why it is different.
Yeah.
That's how
DNA is
mutations occur.
C, D, G, A, and T, and then RNA is G, C, U, and A.
Yes.
Which one was GCAT?
What?
And then the deoxyribonucleic acid ties more so into replication and the gene expression, whereas your ribonucleic acid, your RNA, will help reproduce, translate, and work more so into protein synthesis, which we'll talk about.
That's why you'll see the little lowercase T on the typical
yes.
RNAs.
Little messenger.
So, nucleic acids are macromolecules that exist as polymers called polynucleotides.
Polynucleotides.
I'm sorry about that.
There you go.
So the monomer form, that would be nucleotides, which we're pretty familiar with.
Which are monomer?
They're part of the phosphate group, which is we did
Phosphate's
discuss
their backbone.
Yeah, we did discuss that as one of the most, probably the most important group for life, the existence of life.
Without it, we wouldn't be here.
Yeah, or anything.
What are those 5 elements that we need? It's phosphorus.
It's
carbon, oxygen.
It's carbon, hydrogen, oxygen, nitrogen,
And
phosphorus, and sulfur.
Sulfur?
I think
It's 6.
It's 6.
What do we need sulfur for?
So
I think it's for the bones.
yeah,
It's phosphorus.
It's phosphorus.
Sulfur.
Sulfur is because it's because
I feel like that has to do with plants.
No.
I don't know because I know for a fact that we need sulfur because it's one of the 6.
Really?
Yeah,
That was only 5.
No, there's 6 of them.
It's an animal-based protein. It's in meats, fish, poultry, eggs.
And our fats though,
And plants-based food sources of sulfur are sugar, nuts, Brussels sprouts, and such. It helps reduce, you can have deficiencies in these.
Let's wrap up.
Also, and it's present in living tissue. That's why we need it.
A living tissue.
So,
we basically went over everything.
Yeah,
in this chapter.
Alright, we're gonna go over some of the structures though lastly of that DNA that you find. Or the DNA that we were just talking about.
DNA is usually found in a double helix form, which basically is the most...
Oralia illustrate.
You can illustrate.
I can illustrate.
That's the most thought of image when you think of DNA. It's 2 strands following each other side by side, wrapping each other.
It's a braid, if you will, but with 2. Kind of like a
and the insides are where the what's it called, connect.
The hydrogen bond.
The hydrogen bonds connect, and that's where the different letters come into play.
Yeah. Alright. Here's our drawing. It's not 3D, but it's good enough.
It's a good 2D one.
And the
And these are the hydrogen bonds.
Okay, so as you can see, the 2 sugar phosphate, just keep it in. The 2 sugar phosphate backbones run in the opposite 5-inch to 3-inch directions from one another. And this arrangement is referred to as anti-parallel. And it's similar to like a divided highway. And so the sugar phosphate backbones are on the outside of the helix, and the nitrogenous bases are paired in the interior of the helix. And then the 2 strands are held together by hydrogen bonds between the paired bases. So you can see that within the drawing. I think Meg is pointing to it or trying to, but yeah.
and,
Thank you. It's not a good drawing. It's not good enough for you to point out all those things, but we tried.
And the insides are the hydrogen bonds connected. It's usually a good way to remember is GCAT because G and C will stick together and A and T will stick together. A being a D9 and then adenine, T being thymine.
T being thymine,
Thymine,
G
G
being
being-
guanine,
And C
which
being-
always pairs with the cytosine.
Yes.
Yeah.
GCAT.
And then the G,
G-cat.
and then the C would be cytosine.
Awesome.
And RNA is usually just one strand by itself. Messenger RNA, if you
Or
will, copies.
transfer RNA. They're
Yeah.
Transfer.
different forms.
Yeah.
Messenger,
And it usually
transfer.
just copies one side.
And just a little thing to help you memorize the G-cat. I have John
Ugly.
a
good cat. A
Ugly
good cat.
cat.
A good cat.
It's ugly.
G
It looks like a dog.
cat.
G for good.
G-cat.
Good cat, if you will.
We've got a dog here.
It basically looks like a dog.
It's
so.
a dog. Okay, and I
What's
think
your
that's
chapter?
good for this chapter.
Thank you.
Yes.
Thank you so much,
Thank
Ms.
you.
Gaston. Bye.
Bye.